whole idea is to try and build everyone's skills up so we can just do it consistently, but it's not to inhibit you from experimenting, I guess. It is on-farm manufacturing, so there's always going to be not a sanitised shed and all the rest of it. So I really like the fermentation techniques because I don't need pumps and bubbles and lots of troubles. I always found that the high oxygen bubbling stuff was just more finicky. I'm not saying you don't get a great product, you can get a really good product with aerated compost teas, but they're just a bit more finicky for on-farm manufacturing. And so the bioferments kind of are quite a low tech and easy to make. You sort of set them and forget them. So it's just a bit easier to manufacture, but they're different types of products too, as we'll go through. So that's what this manual is about.
Less risk a little bit too?
I think so. Like, yeah, I mean, both of them, you know, you've got to make sure you've ended up with a stable product. But yeah, with a live aerated compost tea, you've got a very narrow window to get it out. Whereas a ferment, once you've made it, it's shelf stable for a couple of years, most of them, as long as you store it properly. But they are different products, I'd suggest, Matt. So a live compost tea is obviously a broad spectrum inoculant, whereas a bioferment's usually a stimulant plus nutrients, most of them. So the microbes in the bioferment, they're not the main game. So, yeah. You got a question or just putting your hand up? Oh, cool, just waving your hand up. You go, what's he waving his hand up? So that's all good. Now I know what, it's all cool. Yeah, so that's what that manual is. So it's not pretending to be everything. There's lots of stuff on YouTube, and I'm sure some of you guys are making different recipes. But that's what it is, and it's come from a couple of those fermenting traditions. So I just thought we've talked about the 3 techniques, and we're going to concentrate in on the fermentation technique. If you were to ferment anything in a barrel or a tank or whatever, what are some of the things you have to line up to successfully ferment? Whether it's a home brew or a biofertiliser or culturing microbes, what are the key things you've got to get right? Temperature's key. So temperature's one thing. What else you've got to do? The level of air. So some ferments, they're low oxygen or no oxygen. Others are high oxygen, like an aerated tea. So the level of oxygen, and then most of these guys are exclusion anaerobic systems, not all of them. So you want to lock it down to stop the air getting in, which we'll show you up there. So yeah, level of air and temperature. What else you've got to line up to get a successful brew going?
Well, you want to feed it.
So what does everything on Earth need? Carbohydrates. Yep, we all need carbs except people on a keto diet. Everyone else needs carbs. So, sorry?
Line up the planets.
You've got to line the planets up too. I can't help you with that. But yeah, you need the carbs. So microbes need carbohydrates as well, like everything else. But they also need other types of food, including nutrients. So we all need carbohydrates and nutrients, along with air and water, and that's exactly the same for microbes. So on page, which page is it? Page 9 is a bit of a summary of just what you have to get right in terms of a balance. And it's the temperature, the water, the air, nutrients. So you could be feeding them soluble nutrients, or you could be feeding them seaweed meal. But you're giving them nutrient compounds. And then you've got carbohydrates. What's the main carbohydrate source that we're using in these recipes? Molasses, yeah. So the Korean natural farming techniques, Luke uses a lot of sugar, brown sugar instead. And people can use fruits, like a fruit pulp, because it's got lots of sugars in it. But the reason we just stick to molasses is because we're trying to give you guys some consistent recipes at the moment. So molasses for energy, different nutrients, as you'll see, come from different sources. Some out of bottles and bags, others just seaweed meal or fish waste. And then you've got, that's the energy, the carbohydrates. And then the other one is, of course, a starter culture. So when you're making home brew, what do you usually add if you're making beer, for example? Yeast. So yeast does the culturing. So it's the same in these recipes. We want to add a starter culture. So the South Americans were really big on manure, and a lot of the big no-till and the cane farmers that first jumped into it 10 years ago, or more, 2012, some of the first farmers in Australia were doing this at scale. They were using cow manure as their starter culture, and as I mentioned, you can get great products from it, but it's problematic when you get to scale in terms of screening it and filtering it it's just a bit, it's trickier when you get all the suspended fibres and dead dung beetles and all the rest of it. As well as the source material might be variable from batch to batch. So I'll show you a couple of different starter culture options, as well as cow manure if you really like your cow manure. Who really loves cow manure and likes to use it? So if you are going to use cow manure as part of some of these recipes, then make a slurry that you strain and use the slurry. That's how to get around it.
Another factor might be time as well, isn't it? Because your cow manure is going to be great over a longer period of time. But
Yeah. Oh, and look, I'm yet to be convinced that one's better than the other. Like, from a time point of view, the kefir-based recipe is the quickest. It is a little bit more money, but it's much faster than any other starter culture, and it gives you a really consistent result. So that's why we strongly support the Australian dairy industry. Even if I'm a vegan, I'm happy to support it. I'm not really vegan. I eat cheese, but I don't drink this stuff. But yeah, it's good gear, so it makes a really good product. Yeah. So you need a starter culture of some kind. So I'm just going to take you through the starter cultures and some of the recipes just quickly in here. And then we're going to go out and make a batch. And as I mentioned at the beginning of today, because it's such a large group and the shed space and the shuttles are all pretty crowded, I'm not going to break everyone into small groups and get everyone hands-on, which we normally do, like someone making milk and someone getting the molasses organised. But I am going to ask for volunteers who are under 30 that have a fair bit of muscle power to do the molasses lifting and stuff. I've got brain, but
He's on the
so
food.
we're going to need
Some work experience here he is. These boys, I think it's
And if you didn't wear the dirty gear, then it's going to be dirty at the end of the day. So we did a day at Waitchy there, Western Victoria last year, was it? No, earlier this year, and they've got this fancy way of getting the molasses out of the shuttle into the other shuttles. It's kind of like a Venturi system that sucks the molasses and water and then transfers it to this third tank, and the end of it is starting to go like this in the shuttle, and there was about 25, 30, and everyone just went back except for yours truly. And yeah, we got molasses all over us because it just popped out of the shuttle. So you do get messy doing this stuff until you get it organised, and it leads me to the other key part of this whole thing, and that's equipment. So you've got to be organised with your site and your equipment, and your gear, water, clean water. Like, do you need a front end loader for your tractor so you can lift shuttles of molasses and just let it gravity feed into the tank? All those kind of things. We can talk about equipment because Andrew's got gear here from what he's already making, but there are other equipment options, and some of you guys probably can throw into that pot as well. And the last thing is cleanliness. So keeping your equipment clean. Flushing out, hosing down between runs, those kind of things. So there's a bit to doing fermenting correctly, but it's not too complex, and you can get away with it on farm even though you're going to get cross-contamination because there's microbes everywhere. Molasses itself is has a huge bioburden, we call it, so molasses is full of microbes. But the point is, when I chuck them into a certain recipe with no oxygen, only certain ones become active and dominate. So what we're trying to do is use a culture gang that does jobs for us. So the culture is the first really important thing. So just flicking through the recipe. Oh yeah, and then there's like quality control. So page 10, you'll see it's about following the process and monitoring. Unfortunately, because I'm on the road for 2 weeks, or nearly 3 weeks, and I haven't come from home with the ute, I didn't bring the pH meters and stuff like that, so there's bits of gear, there's photos of them, but I haven't got them to sort of show you. But we'll sort of talk you through monitoring and quality control and just keeping things clean, which I mentioned already. The next version of this manual, we're going to have some cleaning procedures and different options. Pool chlorine is fine, all that kind of thing, which you can use. Oh, you got it in there. Great, thank you.
Yeah.
Oh, there you go, you've got the next version of the manual already. So that's kind of, just some simple cleaning at the back there. And before we get into the nitty gritty too, you can use different ingredients. You can swap things out and mix things around depending on what you're trying to do. I need more phosphorus in the paddy, Dave, or I need this and that. You can, but I guess what I'd suggest to you is make a few batches with some basic recipes to get your head around it and get some confidence up. And then you can start to play around and adapt them because you'll have the foundation of kind of principles in your brain by then. And there's no one certified organic here, so we don't have to stress too much about that, but there are options if you're certified organic as well. So with that in mind, there's just a couple of recipes in here, but I'm going to start with the base cultures. So the key thing about any fermenting is you've got a base set of culture that you use to do your culturing, whatever it is you're making. So in these ferment techniques, there's a couple of options for making your base culture. The ones that you make on farm, there's two in the book there. There's the cab serum and the lab serum, which one's based on rice water and one's based on the kefir yogurt. But that's sort of generally what they look like and kind of what they smell like when they're finished. So I'm just going to pass that around. Don't drink too much, Tim, or not everyone can have a swig. We'll just have a look at that. And these guys, the guys here, Andrew and Barry and Co, have made a batch of the starter culture for us to use in the recipe. So I'm going to hand this around, starting with this end here.
Is that the rice?
So this one here, that's coming that way, is the one made on rice water. Correct, Andrew? That's the one you guys have made on rice water. And you'll notice how it's a bit thicker. It's quite thick. That's because we've decanted it from the bottom of the shuttle. So generally, it's not usually that thick. It's usually this sort of thickness or viscosity, this one in the apple juice. That's sort of more what you get. It can be a light brown to a mid brown.
Have you noticed any difference in the final products by which one you use?
That's one of my missions, Matt. Our mission is to actually test. We want to do test manure versus all the starters, because some people will tell you manure is better. What we know is that the lab and the kefir in particular, they just crank off and do a really consistent ferment really well, like each time their batch. And Simon, who works with me, he's a reformed cane farmer from Mackay. So he's set up on his farm a pretty big operation. So he's been doing a lot of batch testing, and he sees the kefir under different times of the year where you get seasonal temperature changes. The kefir is more consistent through the year. But the rice water one does a great job. Like I've used that for many years, and the commercials like BAM and stuff do a job as well.
Yep.
So you can use them. It's just about consistency. Yeah, but I mean, that's what I want to do, a test on the different ones to see, okay, is one really better than another? But what we know so far is, as long as you get a good fermentation kick-off, they go through a good sort of pathway, if you like.
Have you got a
We've
ratio?
noticed a bit of mucking around Duncan and I've done it exactly that. We've got to the end result, we're like, well, hang on, is there much difference there? But at the start, you've either got to leave the lid on one or off one, because one will take 3 days to get going, whereas the next one will only.
The kefir jumps straight away. The one based on this, like, I'll never forget the day we went to see Johnny. We drove about 4 hours up to see him, and he just looked at us, and we started telling him, oh, we're using manure, Johnny, and this and that, and he just looked at us like, what the hell are these characters doing? And he said, do you know, just use kefir, and we went, what the hell's kefir? And we went down the supermarket, and it just does the job.
Yep.
Because he came out of the dairy industry, it's got a known concentration every batch,
Yep.
so you get a consistent concentration of your beneficials, and you get these guys missed it, yeah.
Thank you.
So there are different starter cultures, I guess is the point. I think Andrew's used BAM a bit, and anyone else use BAM here? Yep, so BAM is beneficial anaerobic microbes, also known as a basic starter. So yes, you could just, some people, if you're busy that year and you don't want to make your own, just this is obviously an alternative you can just buy off the shelf.
I think he's got a recipe NTS now where you can use a bit of that to culture up.
You can expand it.
But
Like you do yogurt.
but I don't know if your cell counts and that's all.
It will reduce over repetition. So if you get 20 litres and culture it to 1,000, you'll lose a little bit of concentration. If you then take from that 20 litres and culture it, you're going to lose a little bit of firepower. But it's an option for you.
Have you got a culture ratio for that kefir?
Yes, all the recipes
Oh,
in the book.
sweet.
Yeah, the book's got everything for you. Yeah. Basically, don't drink more than one yourself while you're making it. So, and I'm saying that because there's a couple of cane farmers that up around Tully in North Queensland that make this kind of stuff and put it out at boiling on the sugar cane. And there's three brothers, the Otone brothers. The first time they ever made a batch, they lived in Mission Beach, the tourist town. So they went to Woolies and cleaned it out. And the older brother was going, Mick was just drinking it, and he got a really sick tummy, and the other brothers were just laughing, they were on the ground laughing. When I went to see them, they said, and Mick said, that stuff gave me a sick tummy. And I said, how much did you drink? And he knocked back 1.5. So you don't have too much because it's pretty strong.
Some of the guys in the tropics were using tomato paste. Have you done anything with that?
I haven't, but we have, making the hydrolysate on food scraps. Yeah, for sure. And there's a recipe in there that you could put tomato paste in. The cane guys, I was lucky enough, done about 10 years extension up there around soils, and some of those cane guys around the wet tropics, Tully to Cairns, they were really early on this stuff, and they went to South America, a couple of them, to see meet the Masumas and meet the farmers.
I was privileged to do a bit of work with Chris Nathaniel. Tropiculture,
Yes.
out of Darwin. So
yes.
Wealth of knowledge. Wish I could download his brain.
Yes. No, there's quite a lot of innovation up there because it's so far away. Not everyone sort of hears about it. But the cane guys up there, like a lot of them are kicking big goals. But they got warm temperature, and molasses for them is as cheap as chips. And also it's all warm, so it's easy to pour and pour out. So they have to heat it up and all this kind of stuff.
And they're all privileged in making alcohol, which is not much difference.
So it's a kind of it's a good fit for them. So I guess the key point is there's different starter cultures. In your book there's two that we've just stuck in there. One is based on rice water. Page 13 is one of the tropical fruit growers, Bob Bright, who makes a lot of bioferts for his bananas and sourshops. So page 14, I think, is the lab serum. So lab serum is a traditional culturing starter culture on page 14. And all you need for it is rice, water, and milk powder and molasses, basically. So the guys have made a batch. That one coming around in the 5 litre jug is made on this recipe. And it's a starter culture, a basic starter culture, and all you need for it is rice, water, molasses, and milk powder. So Jerry Gillespie, who's been another mentor of mine, he's a bit of a compost and biological guru. I'm not sure if his picture's in here. Anyway, Jerry, that's the go-to recipe he uses for his fermentation composting that he developed. So it works really well as a starter, and if you're in a country where you can't get kefir and there's no supermarkets, you can usually get rice and milk powder. So that's why it works well. So that's your starter culture. And then the second starter culture, which I'll just introduce you to, is the one on page 16, which is the kefir one, 16, 17. So the key thing about these recipes, the kefir one, the one made with the yogurt from the supermarket, is that it's just quicker. The rice water one it's a couple of extra steps, so it takes you about 2 weeks to go through, or 10 days to go through the steps. The kefir one is literally a one pot mix. You just mix it all in the shuttle and leave it, and it will turn into your starter. The downside is, of course, you've got to get the kefir yogurt. So some people that live fairly remote, their supermarket doesn't sell this stuff. But if you live near, like you guys got this early this morning in Coles in Murray Bridge, so like it's around. They don't have anything around. So normally what you do is you go in and see the person that manages the dairy section, and you give them the heads up and you say, I want 2 weeks' time, can you order more kefir in? Because otherwise I think there's a hippie festival going on in town. Why was there a kefir drunk? So you just go in and order extra, and then it will be, you'll have it there, and they won't stress out that there's no kefir for whoever drinks kefir in town. But yeah, so there's different versions of it. This is babushkas, which is a pretty common one around. And this one here is not called kefir, it's called fuel milk, which is a Scandinavian version of kefir. They call it fuel milk, sour milk. And it's also applicable, and you'll see in there, I've mentioned it in there. I lived in Sweden 3 years, so I used to actually drink buckets of this stuff, but it's very similar to kefir. So both of them work well, and there's different brands. I'm not promoting these two brands on their own, but that's just was available in Coles this morning. The good news is we've just done some recipe testing with the ratio of the kefir, and you can halve that rate. So we're pretty confident now you get a good starter culture with less kefir. When we originally developed the recipes, I just wanted to make sure that they would guarantee to give you a good starter, so we upped the rate. But you know, no one could tell me what the concentration needs to be to get this many microbes in the starter culture. So we've just done some batches at half rate of kefir in the cap serum recipe, and they've come through really well. So if you want to put that full rate in, feel free to. If you want to do half rate, you can. And what you'll see when it's really cranking, it'll look like Coca-Cola bubbling in the tub. Like it really fires up. So, but you could go in at a lower rate of kefir and fuel it up. So you've got rice water and the lab serum based on rice water and milk, which will show you. The guys have made a batch and you've seen the end product. This is a bit thick than normal. That's just because it's come out of the bottom of the shuttle. That's why it's a little bit thicker, which leads to the point that you could
Had a few problems with the curves.
get. Yeah, right. Yeah, we'll go through that maybe when we send the shuttle. That's the thing about the rice water based recipe. There's a couple of steps to it and it's a bit more finicky, but it does give you a really cheap, effective inoculant. Or if you just want to buy an off-the-shelf one, you've got a couple of options if you haven't got time to make it this year yourself. Because they sell this in 1,000 litres, I'm pretty sure that they have seen 1,000 litres. So does that kind of make sense to everyone with the starter culture? Any questions around the starter culture?
I think that's handy when it goes up and almost the same before the rest is made it from one.
You can expand it out, yeah. The question is, if you do that too many times, you will lose some of the potency of it. So that's another thing we'd like to do trials on, is make a cab-based serum or a lab-based serum and then try and just take some of it and culture it on. Yeah, but I'd like to do, and I'm only being particular about this because I've learnt from Chandra, who does this commercially for a living, that you start to lose concentration of your beneficials. So I bang on about consistency, because if you're going to put in 100,000 litres of tanks and set up for this, I want you to have a good product each time as best you can. That's why I bang on about consistency. But I think there's potential to do that to save a bit of dollars. Yeah, it's just testing out how much potency we lose, and is that a big deal anyway. But we're pretty confident you can lower your kefir ratio and get a good result, because we've just done a batch test. Yeah.
Is there a critical mass? Can you downscale it for a trial?
Yeah, so you can do it 20, 30 litre home brew scales. You know those 30 litre?
Yeah.
That's what we do our recipe testing at. So if you just want to go to the home brew store and get the 30 litre all-in-one unit, it has a thermometer sticker on the side and a tap, it's easy. Just do a batch test in that. There's a 200 litre ratio recipe for 200 litre barrels or 100 litre barrels, and then there's the 1,000 litres.
So just divide the 200 down.
Correct, by 10, yeah. Is anyone doing larger scale than shuttles? Has anyone gone to 7 or 10,000 litre tanks?
Yeah, we've got a 5,000 litre tank.
Yeah.
So the 2,000 litres, how many hectares does that do?
Of a product, or which product? The starter culture.
Yeah.
So the starter culture goes into your other recipes at different ratios, but it's usually around 10%. So if you've got 1,000 litres of a starter culture, it'll usually get you, 10,000 litres of product. Depends. If you have a look at the different recipes later on, you'll see they usually use around that 100 litres per 1,000 litres. 100 litres of starter culture for 1,000 litres of Biofert or whatever you're making.
And then you're putting it out at different rates depending.
Correct, yeah. I mean, the good thing, we'll talk about, like the Sabo, we'll have a bit of a Q&A about using this stuff, but it doesn't burn crops, so it won't burn your crops. So does that starter culture sound pretty straightforward? You've seen what they look like. So the key thing is the pH will drop to under 4. So the starter cultures, the pH drop. So you can clean your ring, your jewellery in them. You can get rid of dead bodies if you're in the mafia or whatever. So they will, they're not that strong, but they will clean your ring. So the pH drops, and that's how you know, along with the colour and the smell, that sweet to neutral smell, the colour should be, the pH should drop to under 4 and under, very quickly, within a couple of days. It should drop. And that tells you, and that will be shelf stable. Those starter cultures are shelf stable. I've had them in Albury. I've had a couple in Albury for more than 3-4 years. And then just store them nicely in the shade, do up the lid once they're stable, and they'll be shelf stable.
No temperature control. and
No,
stuff,
no. They were in a shed on the Tay farm, going through summers, before the last 3 cool years. They went through a couple of really hot summers. And then we still use them to make the tape students. I was getting them to make 200 litre batches of different things. So they are pretty shelf stable once they're made. You can use them to clean as part of cleaning, natural cleaning products and toilet cleaning. There's a whole heap of things you can do with it. Anyway, if I go down that track, I'll get into trouble. So, okay, so that's the starter cultures. You can kind of see how easy it is. We'll go out and show you how to sort of put it together at large scale in a minute. But I'll just talk you quickly through the other recipes, and then we'll get out and make a couple of batches before lunch. So the rest of the book's got recipes in it that use a starter culture. So you get these starter cultures, they're shelf stable, I can leave them somewhere and just use them as I need them. And then from them I can make all kinds of products. So in the book there, you'll see on page 18, one of the things you can make with the starter cultures is a compost inoculant. I won't go through this today, but if anyone is composting, then you can do a kind of fermentation minimum turn type composting and use an inoculant that you make with these starters to help you with that. So if anyone is interested in that, have a read of that section. Page 19, you can see the compost inoculant picture of it in colour. But that, I won't spend any more time. Is anyone here composting? I know I was talking to you, Leon, about it. So yeah, if you're not aware of the fermentation method of composting and you cover it, you save about 80% of your water and a lot of your labour. I've got some fact sheets and there's some YouTube videos on it, but it's worth having a look at. I'm not saying that turn composting is not good, it is, but there is another technique you can use which is called, we call it fermentation or spice compost method, and this is the inoculant you use for it. So the next recipe in there on page 20 is the hydrolysate. Hydrolysate is a bit of a technical term. Basically, it is anything with a lot of protein that you ferment. So protein, high protein fermentation. So food scraps can go in, fish, meat, blood, kelp, coffee, coffee grinds, you name it, it's quite high protein coffee grinds. So anything that's relatively high protein that you smash up, you can then ferment. So the recipe is quite straightforward. It's just water, starter culture, molasses, and your material. It's a pretty simple recipe. So, has anyone used fermented or high fish emulsion or anything like that? Where are you getting the fish from? South Australia, I'm assuming?
The Murray River.
Yeah, where's it coming from? Sampi?
Sampi.
Yep, so how do they make that stuff? How do they make it? What do they do?
Heat it, boil it,
Yep.
and then they'll strip stuff off it.
Yep, so they render it down and then you get it as an emulsion or you can get it as a dry powder. So how are you guys normally getting it? As a liquid emulsion. Yeah, you will get fish powders as well, but you're getting a liquid.
Fishing?
Fishing.
Fishing? You go fishing first? No?
You can go fishing, but we just buy it straight from the commercial fisheries. Charlie carp, obviously that's an emulsion type product where they've macerated the fish up into a suspension almost. So the key thing what we're doing with this recipe is we're taking that fish material or vegetables or blood, you can use blood from an abattoir, and we're putting it through a microbial fermentation process as a second step. Unfortunately I didn't bring you because I've got examples of these finished products that I could have shown you, but I put it in so I couldn't bring them. But the end product usually won't smell very fishy at all if it's gone through a microbial. It actually smells quite stable. What might be the benefit, the extra benefit of putting that through a microbial fermentation? It's already a good product, but why would I then bother to put it through a microbial fermentation?
Make it plant available.
Yeah, well it's already got amino acids in it, so it's already pretty good in terms of that, but it could help with that with a bit. But the key thing that microbes do in a fermentation is they make lots of byproducts. So they make lots of byproducts that we call secondary metabolites or fermentation byproducts, and most of them are biostimulants. So the real benefit of fermenting with microbes is you're actually going to make not just the fertiliser side to it, but you're adding these extra compounds into it which are biostimulants. so for plant stress and things, as well as helping with uptake. So what you get at the end of a properly made fermentation is nutrients in a bioavailable form, but I'm also getting all these compounds that are biostimulants. And that's why the fermentation with the microbes can add this extra dimension to your products. That makes sense to everyone. So fish hydrolysate's a great product, fish emulsion. But if you can turn it into not just a great biofertiliser but add biostimulant value to it, then it could help you, can help you in some other areas of your agronomy.
With those emulsions, when they do strip a lot of stuff off, they are just replaced with synthetic base. Some of them with synthetic nitrogen, but not whereas with the hydrolysate, we've just left everything in there, so it's all in there, you've got a bit more organic matter in it. Yeah, and having that diversity of compounds improves the quality of the end product and the range of biostimulants you're going to get as well. So I mentioned that those international congresses and all the researchers doing it, I've even seen scientific trials where vegetable-based hydrolysates, the amino acid profile is different than out of, say, meat, and that can affect plant physiology. So, like Joel Williams talks about, different amino acids influencing different plant functions, and they've seen that. I couldn't believe he hadn't heard of those biostimulant congresses, because those guys have been researching exactly that. Yeah.
So some crops, for example, respond better to a vegetable-based hydrolysate than a meat one, and vice versa.
Joel's got a good one too with his nitrogen side. So blending diluted urea or whatever you're going to use, sulfate of ammonia or whatever, with something like the hydrolysate, even when the percentage of urea or nitrogen that goes under leaf and things like that, being able to really get the best out of all those things.
Yeah, I think there's definitely synergy from bringing the two together. Yeah. I guess the extra thing that these bioferments have in them is biostimulatory compounds. So the end product that you're making has microbes in it, but they're actually not. There's not many there. When you look under the microscope, you go, "This is crap." But that's not why you're doing it. The microbes are helping you make other products. So the end product is not an inoculant. It may have a few bugs in there, but actually it's got a pH of 5 to 4. Most microbes are not really happy, except your fermenters.
Could you give an example on when you would choose to use the, say, meat-based?
Not off the top of my head, but I can send you some of those papers. And if you're really interested in the mode of action science stuff, just type in World Biostimulants Congress, because it's a massive bloody site.
What did you call it, sorry?
World Biostimulants Congress, they call it. And they have a scientific, get together I think it's every second year. But they do, there's lots of papers come out of it. Some of the papers that Joel Williams throws at us have come out of that work. So yeah, it's just an emerging area that's really interesting, because obviously it's going to affect plant health and stress in crop. So that's the hydrolysate recipe. The guys have brought some fish along, I think. What have you got? The Whole Foods South Australian organic fish from somewhere. Yep. Straight out of the national park. Don't say that, Dave. So you guys are going to do a Whole Foods. That's it. We've got a marketing name there, Leon, with fruity notes. Whole Foods South Australian biofood fish. So yeah, we're going to take you through that recipe, and the guys have got the fish, so we'll make a batch of hydrolysate. What's the end product? It's an amino acid based fertiliser, not a high analysis product, but it has this range of biostimulants and amino acids in it, and could really complement a program. Most fish products, natural fish products, like the Charlie Carr one or NTS's fish or Seasol's fish, if they don't have extra nitrogen chucked in on them, they're never usually higher than 3 to 4% nitrogen. So if you want a high analysis hydrolysate that's full of the N, you'd have to put a lot of extra stuff in. So I know Sustainable Farming Solutions, and know where Ben's gone, he's done a runner, thank you. Oh, that's the analysis on the fish emulsion.
Yeah.
Great, hydrolysate, great. And how have you guys broken that up?
So Duncan and I have got a few different brews of works with different products there, so we can effectively replicate any of those ones.
Yep.
It's whether it's due to seasonal conditions or whatever, to what's available.
Yeah,
that's the main line.
Yeah. So are you microbially?
We haven't measured the microbes in there. But
But you've been using the culture to break it down. Right. So it's a microbial fermentation, because a lot of the other way people do it is just put straight enzymes, powdered enzymes in and break it down. You guys are using microbes, yeah. So, and I think this is the key thing. If you use microbes, you're going to get those biostimulatory compounds that you won't get just through enzyme hydrolysis. Because you can just buy bags of enzymes and chuck them in there with the fish and it'll break it apart for you.
Yeah.
But the microbes will just add that extra layer of biostimulants. Yeah.
Is there a rate of microbes or biostimulants within that concentrated liquid level that you get more out of it by spraying? Say, by...
The first challenge I have
Yeah.
is measuring how many
Yeah, I was
biostimulants
just
are
going
in
to
it.
ask, yeah.
Yeah.
So really this is just a
I haven't
spray
found-
and
I
pray
haven't-
approach in the sense that it should
not a
do
spray-
that,
it's
but
not-
we don't
it's-
know how well.
Yeah, it's not a spray and pray too much because we know our nutrient analysis
Yes,
in there.
right.
But how much biostimulant benefit?
Right, yeah.
So if you look at something like sea salt,
Yes.
which is a biostimulant, how does a company like that measure the cytokinin quantity in there? The problem with these organic compounds, like organic acids and plant hormones, is they're really, really hard to measure.
Especially batch to batch.
The lab, yeah, but the labs, there's no commercial lab that I've found in Australia that will do the testing of, it's organic chemistry testing.
Yes.
So I've literally, the Kiwis, I went to New Zealand and did a job with the guys around Christchurch because they're all getting into this stuff too.
Yeah.
They're sending it to India to get amino acids done. There is Macquarie Uni in Sydney will do amino acid, but they're not a commercial lab. Like
So-
if you and me send, we could be 4 months waiting for it.
Basically, they'd just keep going with every batch, right? We'll get 100 kilos
Yeah,
of this
so
kelp and mix it with this.
They
this,
would have
and
done a typical analysis, which is
Yep.
once a year test a batch,
Yep.
which I used to do in the rock phosphate company. Test a batch of our fuels once or twice a year to get a typical analysis. The problem with all the biostimulants is a lot of those compounds are just not easy to measure. So we know from the science that they're in there, like the universities can measure them, but for you and me or Matt to send away and go, how many biostimulants are in, or amino acids, it's not that easy to find a commercial lab. So we're looking, like we're going to Thailand again to actually, there is a lab there that will do it commercially,
Yep.
but it means going to Thailand, so it won't happen overnight, I'll do it at the end of the year.
And even if you want to test your feed quantities, like what you're feeding, so going the next step, you got the weight gains and so on, you want to check your fodder as well, that's got to go into the US.
Yeah, so we actually have found another lab recently, Rachel, one of the young team members with me now, she's found another lab that has a US parent lab that will do amino acid profiles.
So the Weight Institute couldn't measure any
I'm
of
not,
this?
they might be able to do it, but the question for me is if I send a batch off, I want results within a couple of weeks.
Right.
So some of the unis are set up to do it, but it's not a commercial service.
Yeah, okay.
So you could be waiting, I've done this with DNA, and you could be waiting months and months, and then that lecturer gets pulled onto something else, and then their boss says, oh, what are you doing that for? And it's like,
Changes.
yeah, because it's just not set up for commercial service.
Yeah.
So I'm trying to find a lab that will do that. So there is a feed lab, yeah, she just reminded me, she found a feed lab that has a parent US company that reckons they'll be able to do amino acid profiles for us. If that's really the real deal, then that'll be a bit of a breakthrough. So it is a bit of a challenge, yes. So hydrolysate's a recipe that we'll go through and we'll have a look and we'll make a batch today. I'll keep rolling so we can get outside. But basically bring this up with you and I'll talk you through the steps. It's better to talk you through while you're seeing the stuff out there. But I'm just quickly introducing you to the recipes. So then the next batch of recipes are the biofertilisers that start on page 22. There's me doing a batch on tape with the tape students at 200 litre barrel scale, or it's 120 litre barrel. But basically, when we're doing the biofertilisers, we're getting our culture and some extra molasses and milk and all that, and then we're adding nutrients to it. Instead of adding fish or high protein food scraps, we're adding nutrients, and then we're getting the microbes in the brew to chelate the nutrients and turn them into a bioavailable form of nutrient. So you're getting the microbes to do the work for you of processing the nutrients into an organic bio form. So that's really what you're trying to do with the biofertilizer. So there's 3 recipes in the book, where page 23, that's Fraser Pogue's tanks there near Ardmona in central Queensland. But on 24, page 24 is the first of the recipes, and that's a phosphorus recipe. So that's just my marketing terms there. Feel free, Leon, to come up with something better. Biofert P+. So it's just a phosphorus-focused biofert where we get our starter culture, water, bit of milk, bit of molasses, and then we add nutrients, in this case with a strong focus on phosphorus. So we have the ingredients to make that, so we're going to make a batch of that out there. So we'll go through the nitty-gritty of it out there in the field, if that makes sense. And then the next recipe is a calcium recipe, because I know some people focus a lot on calcium. That's the next one along on page 26. And the last recipe in there, which is a micronutrients recipe, or trace elements, whatever you like to call them, is Maxi Micros, I call it. That is got a broad spectrum of micronutrients. So it's an all-rounder. Does that kind of make sense to everyone? So some people like to make just single nutrient bioferts, like just a copper one, or just a zinc one, or just a iron one, or whatever it is, a boron one. This recipe is an all-in-one recipe, but you can change these recipes if you want to focus more on iron or copper or zinc or whatever. The key thing to remember is you just want to keep the general balance between water, molasses, starter culture, milk. When you're mixing, changing things around, keep the ratios fairly similar. If that makes sense. So we'll go through it in detail when we go out there. Any questions so far? Should we get out and make a batch? Yep, everyone's looking really serious, like a tax seminar or something.
We started the soil test too, what we found the last few years. So if you're making this due to what your soil test requires, you think it needs, when you go through a cycle of a year and do your SAP test, you might be surprised at what you actually want to add in if you want to top things up rather than going off the soil test. You'll see what the plant is you can and can't get through that growing season.
Yes. And you know, that's the benefits of having different ones ready to go. You've got a copper, zinc, phosphorus, whatever, and then you can just tank mix as per your SAP test results. The other side, the downside of that is you've got to have a lot of different tanks in space. The other side to it is having an all-in-one. My logic on it a little bit is most cropping paddocks or agricultural paddocks in Australia, the plants are under stress. It's just the modern world, there's no organic matter, the biology is all out of whack from modern farming, whether we like it or not. So giving them an all-rounder just to help that whole system can be helpful. But I think there's also an argument for starting to target the fibres in as well. So it's a bit of what works for you in your situation. But the beauty of it is now that you can make shelf-stable products that will, that you can then use later on and in a flexible way. And you've got biostimulant benefit from it as well. It's got this extra thing on top of it. Yes.
Sorry, I think I missed a step in the process.
Yep.
What do you add the starter culture to?
Shuttle with milk, water and molasses. We'll go out and do it. So you have water, you add milk and you're right.
No, that's the biofertiliser.
This is the starter culture that you use to make a biofertiliser. So you have a starter culture, you've got nutrients, and then you've got an energy source.
Right.
And you have to mix them all together. But that's your culturing gang that does the culturing fermenting for you. And the cow manure based recipes, instead of using that when they're making biofert, they chuck cow manure in.
Yeah. So these bioferts you can store on farm. So say you can brew up a 10,000 gallon, whatever.
Yeah.
And you can use it as needed throughout the season.
Yeah.
Yeah.
Yeah. And most people use them up that season, but most of them are shelf stable and they'd last you the following year if you needed them.
The bioferts as well as the...
The bioferts are pretty shelf stable too. The hydrolysate recipe, which is based on either animal protein or fish or vegetable, that recipe we've developed it to. The reason we have those ratios in there is because it's come from Kansas State University in America that did some work around biosecurity with the foot and mouth disease. So they developed a way to get millions of cows and turn it into a stable, and they studied that by putting it through fermentation you kill the foot and mouth disease. So it was a better way of dealing with all the biomass than just burn it in a big hole. So we based this recipe on that, knowing that it's biosecurity safe. but if you want to add extra fish in and get a high, you can, but the ratio in that has been thought through just to be really safe, to get a really strong fermentation, just in case there's a risk. I mean, fish is a no-risk product, but if you were using dead chooks or something, you were worried about biosecurity or whatever.